All right. Does it work? All right. Well, thank you, Warner, for inviting me, and I apologize in advance that it'll be a bit of a depressing talk I'll give here. I will also talk about some of the research I'm working on the next days as well. But I do think this is really important if you are involved in cancer research. And I will also say, as a disclaimer, I'm not going to tell you, like Warner said, how to figure out if a study is correct or not, because it's really hard. But I think it's just to make you aware of some problems that I believe are in cancer research, and it probably also applies to other research fields. And perhaps just encourage people to think about what can possibly be done, because there are many different parts of groups and people that are involved in these problems, but the best thing is really to sort of start with what you can do yourself, I think. And I think, now I've met most of you, but my name is Daniel, and I'm at the Center for Cancer Immune Therapy at Herlev Hospital, which is in Denmark, just outside of the capital, Copenhagen. And so I'll start talking about a study I was involved in, which ended up concerning problems of reproducibility in cancer research. It wasn't the intention from the beginning. So one of the things I've been very much interested in is the extracellular matrix, the connective tissue in tumors, and how that is remodeled, how it's degraded and changed when a tumor is progressing. And as Warner also mentioned, some of you heard him before, is that an important aspect of this is that some cells in the tumor, they express proteases and they can degrade the extracellular matrix. It happens in most solid tumors. You get a very massive upregulation of MMP, so it's really kind of a hallmark of tumor progression. And therefore, of course, it's also important to know which cells in a tumor that's expressing these proteases. If it's the cancer cells themselves, or is it some of the host cells, the stromal cells, or the immune cells. And that's, of course, important when you want to model a tumor, perhaps ex vivo in a cell culture system, or in a mouse model. You need to know how the situation is in a real tumor. So what we were asked to do when I was at the NIH in the group of Thomas Bugge studying this, we were actually contacted by a Journal of Cell Biology. They wanted us to write a review on this. They wanted to know, well, which cells are actually expressing these proteases. And we figured that, well, perhaps we could do that. We already knew quite a lot about the expression of these proteases, especially in mouse models, but also a little bit in human cancer. And we thought, well, we knew there were some inconsistencies, but if we really carefully looked at all the literature, we would probably come up with some consensus, and we could perhaps explain which cells that were mainly expressing these proteases. But it ended up being impossible, and instead we wrote a piece on problems of research reproducibility in these studies. So I'll just try to briefly go through. Do I have I can see the time there. I have no idea how long this has taken, but I can cut down eventually. So I'm going to take you through some of the things we studied in this review. So it became sort of a meta-analysis, actually. We went through a lot of literature. One of the reasons we thought we could do this was that we actually knew from some mouse studies that it was quite clear that it was not the cancer cells themselves that were expressing proteases. It was the stromal cells. That was, for instance, the case for MT1-MMP. Also for UPAR, as you talked about, it was quite clear in some cases. And one good thing about these mouse studies were that often you would have knockout control. So you would have a mouse that was just completely devoid of the protease, which was the perfect negative control. And in that case, you could perhaps see that, well, the staining is present in the wild-type mouse. It's immunohistochemistry here, but it's absent in the knockout mouse. So it's a specific staining. So then you kind of really trust it, but you can't really do that in humans. One reason that when you do these controls is that sometimes you get a little bit concerned about immunohistochemistry is that sometimes you also see a very nice staining. It doesn't really matter what this is. It is the mannose receptor, but you have specific staining, at least it looks like it, certain cells, and it actually looks kind of how you would expect the staining to look like. But when you do the same staining in a knockout mouse, you see the exact same staining pattern. So it's not a specific staining, at least not specific for the protease you want to stain. It might specifically cross-react with something else that you're staining here. So when you've seen these kind of things, you also become a little bit concerned about immunohistochemistry in human samples because you can't do these controls, and it can be really hard to make sure that your staining is correct. Anyway, what we did to try to come up with a consensus was this. We decided to focus on 5 proteases that are all important for degrading the extracellular matrix in tumors. And we focused on 4 common carcinomas, so in breast cancer and colon cancer, lung cancer, and prostate cancer. And then we just really just mined the literature for everything we could find where they included either immunohistochemistry, immunofluorescence, or in situ hybridization, so detection of the messenger RNA, of one of these 5 proteases in one of these 4 cancers. And we found almost 250 papers. Often they included analysis of more than one type of cancer or more than one of these proteases, so it ended up being even more data sets. But then we excluded some based on different criteria. For instance, if they didn't include any information about which cells that were positive, but just stated that it was expressed in the cancer or upregulated, then we excluded it. Also, if they didn't provide any figure example, then we also excluded it. So we still ended up with quite a lot of data sets, and we hope that that would be enough to show something. Then to keep it simple, we just divided each study or each data set into one of 3 groups if they detected expression just in stromal cells, or expression just in cancer cells, or if it was expression in both stromal and cancer cells. And then we also had some criteria for when to put them in each, because sometimes it wasn't completely black and white. But if we start out here with, this is the results from breast cancer, and now we just compiled all of the 5 different proteases, but that was of course not what we did initially, but this is just kind of to show you that what we really found was that there were no clear trend for any of these proteases. Most studies found expression in both stromal and cancer cells, but quite a lot also found just expression in stromal cells and some only in cancer cells. If we looked at the other ones, it was kind of the same trend. A majority of studies found expression all over basically, but some only in stromal and some only in cancer cells. And we kind of concluded that, well, based on this, we couldn't really say anything about the expression pattern of these proteases. And we tried different things because, of course, you know, the different types of cancer can be subdivided into different histological subtypes. So we tried to do that and didn't really help at all. Then this was actually not what we decided to do, but we were asked to do this for the revision to try to see if it had something to do with the quality of the papers they were published in, but that didn't help at all either. One thing we really realized was that the variations were more study dependent than, for instance, cancer type dependent. And what I mean with that is that sometimes we would find in one study that they examined the expression of a protease in more than just one of these cancers. And in the cases where we saw that, they would always find the same expression pattern. So if they found like stromal cell only in breast cancer and they in the same study also investigated lung cancer, they would also find stromal cell only. So the variation was not within each study, it was just between the different groups and the different publications. Then, as I mentioned, we were looking at both protein expression and mRNA expression. So we tried to divide all of these into the ones that were detecting protein and the ones that were detecting messenger RNA. And then we actually started to see a sort of a grouping here. And what we saw was that it was especially, now we focus on just breast and colon because that's where we had the most data sets. that it was, if we looked at it in cytohybridization, the trend was actually that often it was in the stromal cell compartments that they saw expression. So it kind of separated the studies quite well here. And if we focused then only on the in situ hybridization, we saw that they were typically done with either a radioactive probe or with a fluorescent probe. And if we tried to also take that into account, then we narrowed it down to the radioactive probes. They actually tended to report expression in stromal cells only. So that was sort of the only way where we could find some consistencies if we really narrowed it down to looking at just in situ hybridization with radioactive probes. And then we of course had to look a bit into which studies that were doing these type of analysis. And one clear difference we saw that was that often in these studies with in situ hybridization with radioactive probes, it's quite a challenging techniques, and often that was really the main finding of these papers. That was the basis of the study. Whereas if we looked at immunohistochemistry studies, it was very often a very small part of the study, maybe like figure 1b only, where they just wanted to show something, or the last figure to just confirm something. So you really felt a difference here where the in situ hybridization was much more thoroughly analyzed. And talking to pathologists, they also really, because we consulted a lot with pathologists about this, and they really agreed that often these studies here, they were of very high quality. So we kind of, our own personal opinion was that probably we could trust that most, but we decided to not really focus so much on that, but more to just describe what the differences here were. So just to sum up, protein expression we found mainly in both stromal and cancer cells, but when we looked at messenger RNA expression, it was most often just in the stromal cells. And the next question, of course, was why it differed so dramatically. I already kind of indicated some of the things that we believed in. Of course, theoretically, both of these could be true, that you have, because these are secreted proteases, so of course you could have expression and production of the protease in one cell type, and then secretion and binding to the surface of a neighboring cell type. So theoretically, it could all be true, but of course, another option is that one of these types of studies are just often more right than the other. And one concern that pathologists, they kept bringing up was that this immunohistochemistry is something that basically anyone can do. And it was kind of clear also when we looked at the studies that often it, as I mentioned, it was just a small part of the study, and you kind of felt that it didn't really matter what it showed. They just needed to include immunohistochemistry. And the thing is that if you just add a high enough concentration of antibody and a lot of substrate and incubate for long enough, eventually you will get a signal. And very rarely did these studies have any sort of specificity controls. So I think that's really the main problem with the immunohistochemistry studies here. I already mentioned some of these problems. Other could be that some of these proteases are perhaps quite difficult to detect actually. Also, once they're secreted, they can diffuse out of the tissue and they might be present at very low levels. The different sample handling when you do immunohistochemistry can have dramatic effects, so that's of course also a big concern. And as I mentioned, very rarely were there any sort of validation of the antibodies and their specificity. We also often saw sort of no critical evaluation of the subcellular staining pattern, because for many of these proteases we would know where they would be found, maybe either intracellularly on the surface of the cell, and often we would see stainings that could be nuclear or anywhere really. And it could, maybe they are in the nucleus, I don't know, but if that's the case, I think it would require some additional confirmation when that's not what you expect. And of course, what would really be optimal would be some sort of complementary approach to validate the staining. So perhaps seeing that multiple antibodies would give a similar staining pattern, or maybe even better to have both in situ hybridization and immunohistochemistry, but that was very rarely something we saw. So to make the study a little bit constructive, we tried to come up with some suggestions for improving immunohistochemistry studies. And I already said some of the things. If you can use some sort of complementary approach, that would be great, like multiple antibodies or combination of immunohistochemistry and cytohybridization. That would be so much better than the typical negative control, if it at all was included, which was typically just not having a primary antibody but only the secondary antibody. And that, of course, is better than having no specificity control at all, but perhaps just marginally better than that. And then, of course, this evaluation of the staining pattern would be good as well. One thing we encountered when we went through all of these studies was that we did actually find one paper that was really dedicated to finding out in a very thorough way which cell type in a tumor that was expressing a certain protease. And that was actually the UPA here, this urokinase plasminogen activator, and the expression of this in ductal breast cancer. And the study is, I think, extremely impressive. It starts out with just an observation that staining of human ductal breast cancer samples, expression of UPA is found in the stromal cells and not in the cancer cells. That was done in quite a lot of samples, 31, and in 29 of them there was a positive UPA staining in stromal cells only. They did also, in just like 3 of these 31 samples, see a weak positive stain for UPA in cancer cells. But based on this, it was quite clear that it was mainly stromal cells that expressed UPA. But this was a study where they were really thorough about confirming the staining here. So one of the things they did was to score the intensity of the immunohistochemistry. Then they also had extracts of the same tumors, and they could do ELISA measurements of the same protease. So they saw a very nice correlation with the staining intensity and the ELISA measurement. That's sort of a different type of control they did. But they also included actually 4 additional antibodies that the 2 polyclonal, 2 monoclonal, they all gave a similar staining pattern. And they did 2 types of negative controls, the classical sort of isotype control, where you, instead of having the primary antibody, have an irrelevant primary antibody that's not supposed to stain anything. And they also tried to outcompete the antibody staining using recombinant UPA. So some really nice control here as well. So it was very, very convincing, I would say. But still, they also just compared to in situ hybridization with the messenger RNA and saw, again, a similar staining pattern. So I think based on this, they showed very clearly that UPA in breast cancer is expressed mainly by stromal cells. But then to take it a little bit further, they stated in this paper that they knew that other people had seen different localization patterns. And they were kind of curious why that might be. So just to sort of really clarify everything, they decided to test 5 different parameters if that could maybe affect the staining pattern. So the fixation of the samples and the pre-treatment to demask epitopes and the paraffin embedding compared to frozen sections, because that was also sometimes reported. And then looking more specifically at the antibodies, and one parameter that they also suspected could have an influence was the presence of just low concentrations of detergent in the buffers they were using. And for some of these parameters, they showed that it didn't explain the differences, but they did indeed have an impact on the staining. So they optimized many of these, saw that, you know, if you fixate for too long, you will reduce the staining intensity, and also you need a certain type of pre-treatment to get an optimal staining. Paraffin or frozen sections, they found a similar staining pattern, compared everything here. And then they looked more deeply into these 2 parameters. And with the antibodies, well, first of all, they tested 3 additional antibodies. And at first they saw the same staining pattern, but then they tried to just, for some of them, increase the concentrations up to the highest concentrations that they had seen reported before. And this is not easy to get an overview here, but what they basically found was that once you reach these higher concentrations, you start to see staining of cancer cells as well. Then they looked at the importance of having detergent in washing buffers, because they noticed that in some protocols and some publications there were no detergent in the washing buffers, and it actually turned out that this made the big difference here. So if you didn't have detergent in the washing buffer and in the buffers you're using, you actually started to see cancer cell staining instead of a stromal staining. And this was one of the antibodies that previously gave staining in the stromal cells, now it stained cancer cells. But kind of just to really demonstrate that which of these stainings is more correct, even though I think they kind of proved it already, they saw that if they now used an irrelevant antibody not supposed to stain UPA, then in the absence of detergent they would also get staining of cancer cells. So it's a completely unspecific staining. So based on this extremely thorough study, they really showed, well, first of all, that UPA in breast cancer is expressed by stromal cells. And the reason that some people sometimes see other things is mainly that they did not have any detergent in the washing buffer and that they were using quite a high antibody concentration. And then sometimes also over-fixation of the samples would completely remove the real staining as well. So you would completely change the picture if you did this. And one thing I think is concerning, which when I'm getting to the next part of the talk here, is really that you would think that a study like this, it must have taken so much time. It deserves to be published in a good journal. But of course, things like that cannot be published in a particularly good journal. So it's published in Laboratory Investigations, which is not a bad journal, but it's not a prestigious journal either. But anyway, people in the field, you would think, would notice this paper and take it into account in their future studies. So you could speculate that perhaps it's completely clarified for UPA in breast cancer now. But that is definitely not the case. Since this publication, I found more than 200 studies that have in vitro studied how UPA expression or overexpression in cancer cell lines affect different processes. And you can argue that if the cancer cells themselves are not expressing UPA, then maybe it's not that relevant to study it in vitro. At least I think, the way science should go would be to kind of build on these really thorough studies. So it's not as easy as you would think. Okay, but this was a very specific case about proteases and expression and immunohistochemistry in cancer. And I'm sure some of you are thinking that it's probably just this niche, this small little field that's completely rotten and they don't know how to do science. It's, the field you're in is probably much better, I'm sure. So the next part I'll talk about, that other people think that the problem is broader than just the expression of proteases. And it was actually called the reproducibility crisis that suddenly came up. It really started with this paper that was published by Bayer Healthcare, so this quite large biopharmaceutical company, because they apparently were getting tired of not being able to reproduce findings from the academia and things that were published even in good journals. So at some point they just gathered all the data they had from their own research labs and published it here. And just to sort of update you on what they stated here, because there were some things I didn't even know about biopharmaceutical companies. I've never worked in one. But in these large ones, before they go into any clinical research and try to develop new therapies, then they actually have these validation programs in-house. So if they find something interesting published, before they even consider continuing in that, they start out by trying to reproduce the finding, at least the main findings from those papers. And they also said that, well, within these companies, it was kind of an unspoken rule that it was only in about half of the cases where they could reproduce the findings. So that was, I think, a bit concerning, but that's kind of just what they accepted and they knew it. But the reason they published this was because they felt it was getting worse. So they gathered everything here and they explained that usually in this validation program it was up to 4 people working 6 to 12 months on the project, and they published that they had been working on 57 projects, mostly in oncology but also some in other disease areas. And the main take-home messages, they divided into different groups, but the main take-home messages was that it was more like 25% of the cases where they could reproduce the findings that were published. And then they asked, well, they kind of admitted that often they had to change things a little bit. They didn't do it exactly like it was written in the paper because maybe they didn't have the same cell line, or maybe they were using a slightly different assay. So they said, well, maybe it's our own fault. Maybe it's because sometimes we adjust the assays a little bit. But it turned out that that was really not the case, because in the cases where they could reproduce it, it didn't matter if they had changed the model system a little bit. They felt it was much more like, you know, if it's reproducible, and it doesn't matter if we change things a little bit. And I think they really wanted to state this and bring this out there, because often at least in cell biology, it's kind of, I would say, a bad excuse that when people can't reproduce things, it's like, oh, it's probably because you have different ions in the tap water, or you get your PPS from a different company. And they said, well, that's not what they observed, at least. So it wasn't these like small differences that made the big difference. Then next year, apparently Amgen, they wanted to top this, and they said, well, we have similar observations, but we just, in our hands, it's much worse than that. So they explained that they had over a decade looked into 53 what they call landmark cancer research papers. So this is all oncology, and in their hands, they could actually only confirm 6 out of 53 of these papers, so about 10%. So that's really, really concerning. And they explained that they really did everything they could to try to reproduce these things. So first they tried themselves. If it didn't work out, they would contact the original authors to get protocols and maybe exchange reagents. And sometimes they even went to the labs of the original paper to try to reproduce it. And it didn't really help a lot, because it was still just these 6 out of 53 papers. Then another concern that they brought up, because they said, well, maybe it's just us in the industry. We're too stupid in the academic world. Maybe they know that there's a lot of bad and not trustworthy science out there, but they know how to navigate in it because it's their world. So they divided papers into the ones that they could reproduce and the ones they couldn't reproduce, and looked at how many citations they were getting. And it turned out that the ones that they could not reproduce were getting at least as many citations as the ones they could reproduce. So that was really a big concern, of course, because it really means that if we believe that it's that many that cannot be reproduced, then they are really causing a lot of new studies to be based on these non-reproducible initial studies, which is a huge problem. They list a lot of consequences, and I think maybe I added some as well to this list here. One thing they bring up is that this problem with reproducing these, it's all, you could say, preclinical cancer research, so it's not any clinical research. But one problem this could be related to a reported historically low phase 2 clinical trial success rate, which is down to 18%, which is a huge problem, because once you start a clinical trial, it is a lot of money you put into these, and a lot of them were failing. And then they mentioned, as I said, with all these citations, that obviously these papers must have led to a lot of secondary studies initiated by other researchers. And altogether, this, of course, would really limit progress in the cancer research field, which is not what we want when we're studying cancer. Also, some loss of scientific integrity, which I think has been a struggle for some persons in the field, because once you bring it up there, it's also an easy excuse for funding agencies and for governments to cut down on science because you're not doing good science anyway. So sometimes people have been a little bit careful about how to really bring up this problem, but I think, you know, within this group here, I can bring it up without any concerns for funding and stuff like that. And of course, a lot of economical waste, obviously. And the most terrible thing is, of course, that it does have implications for patients, because it will slow down the development of new and better therapy. Okay, so am I good on time here? Yeah, I think it's good. Good?
We're perfect.
Perfect. Well, you don't know how many slides I have left, but no, I think it is. Yeah, good. I think I don't have any solution for how to sort of turn this over and start improving some of these things. And also, I cannot tell if it's really as bad as these people say. I do think, my own opinion is that it is quite bad, and I think we need to do something to improve things. And the first things I think which is really good that's happening is that there's has become a lot of focus on this problem. As I said, it was called the reproducibility crisis, and everyone kind of reacted to it. Some followed up with other papers. For instance, statisticians said that, well, actually we're also often designing studies in the wrong way, so perhaps what is published out there is not wrong per se. I mean, people probably found that. It's just that if you set up 50 studies, then you just by chance in one of them you'll find the difference you're looking for, and then you'll sort of hang on to that and publish that, but it's really just by chance it's happening. And there are lots of other, you know, Nature and Science and all the journals, they immediately jumped in, and I think mainly what they did was to try to say it's not our fault, it's your fault. We're doing everything we can, which I don't think is true, but at least they did react to it. And one of the first things that really happened was that a lot of the good journals, they did whatever they could to at least make it easier for people to try to reproduce each other's research. So now a lot of the journals, they allow long method sections. Before it was always, you know, to cut down, you started with the method sections, and it was all just referring to an old study that referred to an old one, and then finally it ended nowhere and stuff like that. Also, they often, well, some journals, they want you, if you have a Western blot, you cannot just cut out one band. If you do that, you need to provide the entire Western blot and supplemental material. And they try to make everything a bit more transparent and help, you could say, each other here to help, you know, reproduce each other's research. Then there are some that jump to specific reagents. That's also the problem. It's the antibodies that are so bad, and I think we can all agree on that, but I'm not sure it's the reason for the problem. But there are some suggestions that antibodies, they should be much more standardized than the way they are now, and I think it has become, at least now it's become sort of good practice to use monoclonal antibodies, which are a bit more well-defined than the polyclonal antibodies, which differ from rapid to rapid, basically. But actually now they suggest that all the antibodies should be recombinant. You can clone them and express them, and then they'll be completely similar every time, which I think would be a good idea, but I'm probably not there yet. Okay, but I think this really brings to the sort of the discussion part about, well, first of all, if this has really become much worse than what it used to be, who's to blame for that? I don't know if it has, because, you know, of course, if you talk to someone who's been in research for 50 years, they will for sure say that it was much better 50 years ago, but I think that's just how aging works. But I think it has become worse. Who's to blame for that? And of course, I think, as I said, you know, we have to start with what we can do ourselves. So the people that are doing the research and the supervisors, of course, they do have a responsibility here, because a lot of it is also about being more self-critical and not just publishing the second you have an observation, but try to also, prove that your hypothesis is wrong, because that's sort of how the scientific method is supposed to be. But the problem is that it doesn't really speed up publication rates, and that is really a huge part of the scientific world now. You have a huge publication pressure. You have it from the funding agencies. You need publications to get funding, to continue in research, and we all want to continue to have a job and be able to buy milk and groceries and stuff like that. You have the tenure committees that hire you, and whenever you want a job, the first thing people look into, because when I have to say I do it myself sometimes too, when I get applications, you get 50 applications, and the easiest thing to look at, that's just the publication list, and just, one side or the other. So there's a huge publication pressure. And one problem, I think, is also the journal policies and the editors of the journals, because many of these journals, they also almost encourage studies that are controversial, and there's really no appreciation of the solid studies anymore. You don't get any credit for a study like I showed you before, where you really make sure that everything in this paper is correct. That's really not what they value. Also, you really have almost no possibility of publishing negative data, even though that's extremely important as well. I thought I had another point here. Maybe it's coming. Oh, it's coming there. Another possibility is actually, or problem is that you cannot, if you try to reproduce another group's finding and you cannot reproduce it, it's impossible to publish that. And I know, because I've been involved in a study like that. It was a cancer cell paper. No, it was a Nature medicine paper, and it was highly cited, and everything was just done to prove that it was correct, and we just couldn't find it. And on the other hand, we actually demonstrated why they saw something different as well. It was impossible to publish. So I think that's a huge problem as well. There could be some higher standards for testing reagents and more detailed method sections, things like that as well. But I think most of the problem really lies in this top part here. This I also already talked about. Then there's this focus on impact factors, and I think that is a problem for many reasons actually. But one thing is really that these high impact journals, they really have a policy where what they're focusing on is sort of a self-sustaining system. They have a high impact factor, and they want studies that can boost their impact factor, make sure that they keep having a high impact factor. And what that means is really that they choose studies, they publish studies that they know will cite well. So that's papers typically in a hot field. They like controversial studies if they find something that's completely new, unexpected. And it's even, I think, quite concerning here that if you look at the scope of Nature, one of the most prestigious journals, they say what they publish is, it is peer-reviewed, which is good, all fields of science, and it's based on originality and importance and interdisciplinary interest and timeliness, how hot is it, they're very honest about it, accessibility and elegance and surprising conclusion. There's nothing about, that the studies should be thorough and well-controlled and reproducible and, correct. So I think I do think that the journals, they also have a responsibility here. There's been a lot of concerns about the impact factor. There was this DORA initiative. It was started at, I think it was in San Diego at what is it called? What is it called? University in San Diego. It's called UCSD. It's called. Where they invited all scientists to sign this thing that there's too much focus on impact factor, and I think what you cite is really that the content of the paper is more important than the journals are published, and everyone cited that. I'm not really sure how much it helped. They kind of continued, and one thing that they were very much looking into, that was other metrics because they kind of said, well, people want some metrics for deciding if a publication is good or bad, so maybe at least we can come up with the better metrics than just looking at the journal they're published in. So they propose different metrics where you look at the individual papers, how well are they citing, how much attention are they getting, and stuff like that. But I think that one concern about that is, like I just showed you before, is that the number of citations, I also don't think necessarily that's a good measure of the quality of the studies. It's a measure of how much attention they're getting and how hot they are, but it doesn't really say anything about how correct and how solid the studies are. And they also looked at other types of metrics, like, how much is it being tweeted or whatever it's called. I'm really bad at social media. So I have no idea. I think the problems would be exactly the same, that it would just say something about how hot the paper is and not if it's good or bad. Okay. I don't know what can be done. I kind of brought up some people to blame for it, and I think that, of course, we should start out by looking at what we can do ourselves. And perhaps, for people here, a lot of you, I know you're teaching, so that's a good opportunity to kind of, implement a better scientific culture in new researchers. There's been some really interesting initiatives, like this one called the Reproducibility Project, and it's sort of a non-profit thing. It's something called Science Exchange and Center for Open Science that attracted, and they got a lot of funding for specifically trying to reproduce some high-profile papers published from 10 to 12. And when they do this, they give the money to a lab and they say, well, here, you're perfectly equipped for reproducing this study, so you get all this money for doing this, and then we publish it. It's a scientific journal where they very thoroughly explain how they try to reproduce every single experiment in the paper. And it's really interesting. You can go in there and read it. The hard part is that they're not going to give you some summary and say, yes or no, we could reproduce it, because what they really encourage is that people have to be critical. So you have to go in there and read the report and see, do I think that this qualifies for that they could reproduce it or not? And often they end up, I think they've done like maybe 10 papers now, and there are 10 reports out there. And it often ends up with something in between. So it's like, well, this thing we could reproduce, this we couldn't, even though, and then they explain we tried to do this and this and this and this and this and this. But in general, I will say it is a quite depressing reading in general. Most often, the main findings they can actually not reproduce. So it kind of confirms what the pharmaceutical companies, they claim, that it's not all good in cancer research. Okay. What we can do, I think, is limited. I already kind of said it, but I think at least once you are aware of this, you can read the literature very critically, because at least you can try to maneuver in this whole jungle of publication and try not to be the one that's basing your own research on something that's actually not very solid. So you can do that by maybe reading very critically. Sometimes you can figure it out, sometimes it's hard to figure out if you can trust it or not. But also, even though it is a waste of time, you could say, I really think it's important sometimes, if you want to base your study on something that's published, to at least try to reproduce like the main finding that you're trying to build upon. But of course, the critical publishing is really how we can also help the world. So if we try to make sure as well as we can that what we publish is actually something others would also find, that will help a lot. There are some like mistakes I think we should try to avoid, like the Texas sharpshooter mistake, if you know that one. It's this guy who shoots first and then draws the bullseye after he shoots. So which in the scientific world is more like, doing everything, and then after that you look and you find something, well, here I have something like this one, this data says I'm right. But the true scientific method is really that you generate a hypothesis and then you try to disprove that hypothesis. Just not to keep it all negative. I think despite that there are some problems, there's definitely still hope. I think it's still extremely exciting to be in research because often there are these big breakthroughs and you can't really discuss that. You have the gene editing with the CRISPR and it's really here for staying, and I'm absolutely sure it's not an artifact. It's true, CRISPR works. There are all the stem cell reprogramming and the generation of artificial organs and the synthetic cells, and of course from our own field, the whole generation of immunotherapy has been just the most exciting thing for cancer research in many years. Even if you haven't been into immunotherapy, just seeing that something based on basic research will end up in a therapy that's really helping a lot of patients is really rewarding. So there's still hope, and I hope that you didn't lose it completely based on this. So, thank you.
Good questions? Daniel?
Yes?
You mentioned something for cancer research, so specifically for cancer research, but you didn't mention that much about this cell-to-cell heterogeneity. So how could this impact in one patient, in one sample, will have different frequencies of subpopulations, and next patient really different frequencies?
No, I think that's a huge problem. I don't think it accounts for any of these problems very much because all the studies from the pharmaceutical companies, that was all what they call preclinical research projects. So things that were generating hypotheses that they would then move on to clinical testing and stuff like that. But I think cancer is, in many ways, it's not an easy disease to study, and I mean, regardless of how thorough people are, I don't think we can completely get rid of these issues. There will be, exactly like you say, just by chance you have, a certain part of the biopsy, it's just very different from a different part, or anything like that could influence. So, I don't know how it is in other fields. I think cancer research field is a difficult one in general. Yeah?
One comment and one question. First one, for my students who are watching the live streaming, I think you forgot to mention the students, because some of the experiments are being run by students.
Yes.
They want to get their degrees, and I don't know.
Yeah, I counted them in under investigators because I'm just sort of taking everyone who's actually doing the lab work. So that's the students and PhD students, postdocs, and everything.
And the second thing is in my field where you can basically share your programs or algorithms. There is this thing that we saw last week, Docker, which is like a kind of environment where you can basically pass to other researchers the libraries, the versions, everything. But of course, this is a programming world. I don't know if it's something that would be possible for a wet lab, that you make a small box where you put reagents, and you put your reagents, your population, cell population, and then you can buy that stuff and you can send it abroad. And then, I don't know, it's just an idea. I know it might be crazy, and passing that between the countries now with the coronavirus.
Yeah.
Possible, but it might be something that we can take from the software world to the
Yeah.
cell biology world.
No, I think that's a great idea, and I think in general, well, I'm not sure if it solves this, but it's definitely not going to make it worse. But in general, I think that cancer research could benefit from a more open environment. The competition is slowing down things a lot, that people are keeping their observation for themselves for many years because they need to publish and they want to, if you want a good publication, you need 30 supplemental figures, right? So you just need to add so much data, which is really often, I think, noise. I mean, I would really appreciate if people would publish their main observation and that's it. So I think in many other fields, I think they're doing way better. Like in physics, they don't even have journals in the same way. They have a repository where they just post their paper, right? And everyone can read them, and it's actually more of a dynamic thing. You can then comment on it, and you can add more data because, that's a good idea. Let's try to do that, and then add. I mean, I think there are lots of things that could be improved, but I think it's going to be very difficult to change, because now we have this system and it's how it's all built up. It's like a game almost with getting the publications, and everyone knows it's a game. You try here and you go down, down, down. You have no idea. You know, sometimes you end up here, sometimes here. It's not like you think one study is better than the other. It's just random. So it's, yeah.
Daniel, I have a comment more than a question. A couple of years ago, I think, or something like that, there was a comment from a Nobel Prize winner who was actually criticizing the fact that journals tend to publish based on mechanistics, and you have to include, it's like almost a single paragraph is an experiment itself.
Mm.
And he actually said kind of something that I took my attention. He said, build houses of brick, not mentions of straw, because the journals want everything completely packed in the paper, and perhaps then the experiments are not well performed, because they need, you know, as researchers, that you need to include as much as possible in order to get published on that high impact journal. What's your thinking?
No, I think I really agree. I think it's a huge problem, and I think that's also what I mean a little bit by the critical reading. I think often it might be kind of a solid initial finding, and which is really supposed to be the main thing of the paper, but it ends up being completely blurred by all these additional things that's added and maybe are not as solid as the main finding. So I think it's a huge problem. And I think, well, I can't remember his name, but I heard him too at a talk, and I think he was really onto something. He's only himself publishing in, there is actually one of these repositories now, bioarchives. So he's publishing either there or in eLife, which is a new journal which he likes because they have a different type of review system. It's very transparent. You can read the reviews. They also say that they were kind of tired of having 3 reviewers. They were each asking for 25 additional experiments. So they actually have an editor that goes together. They have a phone meeting with the reviewers, and they agree on the consensus experiments that they need, and then they just ask for that. It's like gather it all together. So they have some interesting initiatives to at least make the whole publishing system better. So yeah.
And I think I see that there's another problem. I remember Dr. Michael Bloch, when we were at PRIS-Lab, all the time saying that sometimes people are performing experiments without knowing the question. And they want to include state-of-the-art machines that bring very fancy and good-looking figures, and perhaps they don't really know how to control them properly, so you end up giving data that are perhaps very beautiful but not really true. But you could perhaps do it better in a much simpler way. That's also a thing that sometimes you see. I mean, they want to include a lot of fluorescence, and you could do it easily with something simpler that is much more robust.
No, I completely agree. And I usually say that, it's all about big data now, but I really like small data. That's I don't think it should be underestimated. So, no, I agree.
Any other questions?
Well, I have like a comment/question. What do you think about, like, from the point of view of students, how does one not fall into the Texas sharpshooter fallacy when your job, you could say so, is online, and they might say like, oh no, you're doing it wrong if the tests are not right. I mean, how can a student avoid that, but at the same time, I don't know, lose his job?
I think it's a really good question, and I think usually I'm lucky to teach Danish PhD students, and lucky in the sense that I can easily say to them that it's not going to ruin their life if they don't get the publication. But I know in some cases and in some cultures, and depending on which environment and which country you're doing your research, it can really have a huge impact if you get that publication or you don't get it. So I really understand the publication pressure, and I don't know how to avoid it. I mean, I think a good way to kind of promote it, if it's cancer research, like the good scientific practice, is really to remind people that even though you're just working with a cell line in a lab, it will affect real cancer patients in the end. Because I think if you kind of try to remember that once in a while, maybe it kind of keeps you from being too biased in what you're actually doing. But I don't have a really good answer. It's a good question.
I think the problem gets even worse when you are a student and you know that perhaps this is not the right experiment or the right results, but your professor wants you to go further, you know, and then you kind of feel obliged to keep on going even though perhaps you don't feel comfortable.
Hmm.
Any other questions or comments? Well, then I think we have to